Folk remedies for healing wounds by covering them with moldy bread or fungi may make scientific sense. Researchers have now demonstrated that certain fungi have wound-healing properties. Paul Hamlin and his colleagues at the British Textile Technology Group in Manchester hope to develop fungus-based surgical dressings that accelerate the healing of wounds, as well as shielding them from infection. Although no tests have yet been done on real wounds, experiments on cultured human skin cells have been encouraging, he says. Instead of producing antibiotics, as do penicillium molds, the fungi used owe their effectiveness to chitin, the major structural polymer in fungal cell walls, and a sister compound called chitosan. Hamlin says the dressings may even reduce the need to use antibiotics. Overuse of antibiotics is now a major problem for hospitals, because bacteria can become resistant to them. Working with Richard Schmidt of the School of Pharmacy at the University of Wales in Cardiff, exposed human fibroblast cells to 6 fungi strains. Fibroblasts play a key role in wound healing, laying the foundations for scar tissue to form. The researchers used mycelia, the filaments produced by fungi, and found that those that were richest in chitin and chitosan triggered the fastest growth of fibroblasts. The mycelia also appeared to act as scaffolding, attracting fibroblasts and anchoring them, ready for the next stage of wound healing when collagen scar tissue forms. Phycomyces blakesleyanus, a common mold that has been found growing on stale bread, human feces, dead wood, and horse dung, performed best. Of all the molds tested, it contains the most chitin and chitosan. Together, these account for 90% of the weight of the mycelia. It spurred the fastest growth of fibroblast cells, with 17% more cells formed after 6 days as in a control culture. When is oxidized, it generates hydrogen peroxide, and Schmidt believes that this is the secret of the mold's success. Although hydrogen peroxide is toxic in large amounts, trace amounts are essential for healing. The compound activates white blood cells, which fight infection at wound sites. It also triggers the proliferation of fibroblasts, says Schmidt. Hamlin has patented processes for growing the mycelia in bioreactors, cleaning them and making them into surgical dressings. He has produced absorbent sheets for superficial wounds and absorbent gauze for deeper ones. He says that it is possible to strengthen the material by mixing the mycelia with wood fibers. The London-based British Technology Group is hoping to commercialize the researchers' patents, and is seeking industrial partners to develop the dressings. Hamlin says that because the fungi are pharmacologically active, the dressings will have to undergo full-scale clinical trials. He expects them to be of most help in treating persistent wounds, such as pressure sores and diabetic ulcers. The protective effect of carotenoids, polyphenols, and estradiol on dermal fibroblasts under oxidative stress. Skin aging is influenced by several factors including environmental exposure and hormonal changes. Reactive oxygen species, ROS, which mediate many of the effects of these factors, induce inflammatory processes in the skin and increase the production of matrix metalloproteinases, MMPs, in dermal fibroblasts, which leads to collagen degradation. Several studies have shown the protective role of estrogens and a diet rich in fruits and vegetables on skin physiology. Previous studies have shown that dietary carotenoids and polyphenols activate the cell's antioxidant defense system by increasing antioxidant response element, NRF2, NRF2, transcriptional activity. and reducing the inflammatory response. The aim of the current study was to examine the protective effect of such dietary-derived compounds and estradiol on dermal fibroblasts under oxidative stress induced by H2O2. Human dermal fibroblasts were used to study the effect of H2O2 on cell number and apoptosis, MMP1, and pro-collagen secretion as markers of skin damage. Treatment of cells with H2O2 led to cell death, increased secretion of MMP1, and decreased pro-collagen secretion. Pre-treatment with tomato and rosemary extracts, and with estradiol, reversed the effects of the oxidative stress. This was associated with a reduction in intracellular ROS levels, probably through the measured increased activity of NRF2. Conclusions. This study indicates that carotenoids, polyphenols, and estradiol protect dermal fibroblasts from oxidative stress-induced damage through a reduction in ROS levels. Keywords: NHDF cells, antioxidant response element, NRF2, NRF2, apoptosis, carnosic acid, collagen, lycopene, matrix metalloproteinase, MMP, reactive oxygen species, ROS.